Okay, so my name is Megan Butler, and I'm a graduate student in the Department of Molecular and Cellular Physiology. And today I'm going to be giving you a lecture on Chapter 7, which is going to focus on the neuromuscular junction and neuromuscular transition between the nervous system and skeletal muscle. And then Chapter 8, which goes into details about smooth muscle contraction and function. Here are your learning objectives, and you can read through these on your own, and I will be touching on these throughout my presentation. And so the neuromuscular junction, exactly what it sounds like, it is going to be the physiological, that functional connection between the nervous system and muscle. And so this first part again is going to focus on that connection with skeletal muscle. So just to orient you with what we're looking at, here's your spinal cord. Now motor neurons, which are going to innervate our skeletal muscles. Motor neurons are going to leave that anterior or ventral horn of the spinal cord, exit out through the ventral nerve, spinal nerve, and then go innervate the belly of skeletal muscles. Now the first concept I want to introduce you to is that of a motor unit and what that is. It's composed of a motor neuron and all of the skeletal muscle fibers that it innervates, that it connects to. Notice here these two motor neurons are color-coded. You have a purple and a red so that you can see the difference between motor unit 1 and 2. This purple neuron is innervating 2 muscle fiber cells, whereas motor unit 2 is innervating multiple or more skeletal muscle cells. And so what this is, the point I want to bring up here is that there's an inverse relationship between the size of the motor unit and the degree of control, meaning the fewer skeletal muscle fibers that are innervated by a single motor neuron. So the fewer skeletal muscle fibers innervated, the greater the degree of control. So think really small motor units are found in like the muscles of your eyes as well as the muscles of your hands. You have really fine motor control in your fingers and your hands. So you're going to have one motor neuron only innervating a few muscle fibers. So you get really fine-tuned control. Whereas in muscle groups that you need a lot of power, but you don't need a lot of coordinated control, you can have larger motor units, say your quadriceps or the muscles of your back. You can have a single motor neuron innervating thousands of skeletal muscle fibers. So that's the point I wanted to make here. This is just another depiction, another cartoon of the neuromuscular junction. Here we have our motor neuron. And I'll bring up a few points in my next lecture of the central nervous system. I'll go into more details about this. But here's our motor neuron with the little dendrites that are going to receive signals from neurons coming down from the brain about what muscles need to fire. That information is then going to travel down the axon, which conducts action potentials down to these axon terminals, okay? And if you notice in this cartoon, there's going to be one axon terminal innervating one skeletal muscle fiber. And so you can have one axon that can branch, but you'll have only one, in general, one axon terminal innervating a single skeletal muscle fiber. So just another cartoon of the neuromuscular junction. Here's another one, and we're going to go into a little more detail about the anatomy of the neuromuscular junction. So here's our myelinated. Most of these motor neurons are going to be large fibers myelinated so that they can conduct impulses very quickly to the skeletal muscles. So here's our myelinated axon coming down, and then we have a synaptic knob or axon terminal, potato, that's going to come and innervate a skeletal muscle fiber cell. So what I want to point out here is in that axon terminal, if you look inside, there's all these there's lots of these little vesicles. These vesicles contain the neurotransmitter acetylcholine. So when we're talking about the neuromuscular junction with skeletal muscle cells, the neurotransmitter involved is always acetylcholine, and they're contained in these synaptic vesicles ready and waiting to undergo exocytosis to dump that neurotransmitter into the synapse. You'll also notice that the axon terminal has tons of mitochondria, and it's because the synthesis of acetylcholine to make so much of it requires lots of energy, so lots of mitochondria. So that's the synaptic knob, or the axon terminal. Now, the skeletal muscle fiber cell. Here's the sarcolemma, which is just a fancy word for the plasma membrane of a skeletal muscle cell. And this specific area, which is highly invaginated, has a special name called the motor end plate. Okay, so it's a specialized region of that sarcolemma that's highly invaginated. All of those folds means that there's a greater surface area for more acetylcholine receptors to be embedded in that plasma membrane. More acetylcholine receptors means more acetylcholine that can bind, and the greater likelihood that we cause skeletal muscle contraction. So the point of this is to increase surface area for more receptors and more sodium channels as well, and I'll show that on the next slide. Now, the space between the synaptic terminal, or the axon terminal, the synaptic knob, and the motor end plate, this is the synaptic cleft, and that space is where the acetylcholine will diffuse across. It's only a few nanometers, 20 to 30 nanometers wide, and that acetylcholine will diffuse across the synaptic cleft and again bind to those acetylcholine receptors on the motor end plate. So a little microanatomy of the neuromuscular junction. And a little more detail, so this is that same image again, and I'm just going to reiterate the fact that in the axon terminal, you have all of these synaptic vesicles that contain acetylcholine. Acetylcholine is the neurotransmitter, the only neurotransmitter used at the neuromuscular junction with skeletal muscle fiber cells. Acetylcholine is synthesized in the cytoplasm of the axon terminal, but then it's will be put into and packaged into these vesicles, which again kind of lie in waiting, and they're just ready for as soon as they're needed to release acetylcholine. And again, I have some extra points on the slide that kind of help you. You can read through them, but I will not ask you any questions that I don't specifically go over. So if there's 300,000 vesicles present at any single end plate, I'm going to ask more important things in my multiple choice questions. This is just to kind of help you fully understand what's going on. So I have extra information on my slides, but I won't ask anything that I don't specifically go over or tell you that you need to know, so review this on your own. All right, so this, again, reorient you. I know up here, this is my axon terminal because I see all these synaptic vesicles that are containing the neurotransmitter acetylcholine. This is my motor end plate because I see all of these invaginations of the plasma membrane. And the only thing that I really wanted to highlight here from this image that wasn't shown in the previous images is the fact that in those invaginations of the sarcolemma, those acetylcholine receptors are located near the surface, kind of the neck of these invaginations, which makes sense that these acetylcholine receptors are located closer to the surface, so that as soon as acetylcholine is released into that synapse, it can very quickly diffuse across that synaptic cleft and bind to these receptors. The other channels that we see present in these invaginations of the sarcolemma are voltage-activated sodium channels. And so you've had some lectures on action potentials, and you should be somewhat familiar with some of the words that I'm about to use. But what's going to happen is when acetylcholine binds to its receptor, it causes an influx of sodium and a local depolarization. And if that depolarization is great enough, it's going to open these voltage-gated sodium channels. More sodium will influx into the skeletal muscle cell, causing an action potential. And so just like you saw action potentials that travel along the axons of neurons, the sarcolemma of skeletal muscle fiber cells are also capable of conducting action potentials. And that's how we're going to get skeletal muscle contraction. So these acetylcholine receptors are ligand-gated channels because you have the neurotransmitter binding them and opening them, and then the voltage-gated channels are opened when the appropriate local depolarization is met. few details about the acetylcholine receptors is that when acetylcholine is not bound, so here this would be our sarcolemma, the plasma membrane of the motor end plate, and we have this transmembrane channel. This is our acetylcholine receptor. And when acetylcholine is not bound, it's going to be in a closed conformation. But when two acetylcholine molecules bind to their specific receptor, it's going to open this channel and allow sodium to influx into the skeletal muscle cell. And the reason why it's going to be primarily sodium that's driven in is, well, negative ions won't be permitted because the interior of this channel has a negative charge, so it's going to repel any negative ions. So only positive ions are going to be really allowed through. And the two major cations, or positive ions, that we have, like kind of in the extracellular, intracellular fluid, is we have a higher concentration of sodium in the extracellular fluid, and then we have potassium ions found in the intracellular fluid. Well, there's two forces at play that are going to drive sodium inward. It's going to be that high concentration in the extracellular fluid, and then the second force is that there's an electrical gradient because the resting membrane potential, which is just along the interior, the inside of the plasma membrane, that resting membrane potential is negative. So there's an electrical gradient as well as a chemical or a concentration gradient that's going to drive sodium in when these channels are open. That's going to cause the membrane potential to become more positive or depolarize. And then if that depolarization is great enough, it can lead to the opening of those voltage-gated sodium channels and an action potential. And so that I guess the term that I should highlight here is end plate potential, kind of like a local potential, which you may have learned with your action potentials lecture. But when sodium influxes in through these acetylcholine receptors and depolarizes, that's called an end plate potential, right? If that end plate potential reaches a threshold or is great enough, then we can open up those voltage-gated sodium channels and get an action potential on the sarcolemma. All right, so I think repetition helps. So I've tried to go ahead and mention some of these things, but let's, from start to finish, how do we get acetylcholine onto the receptors and an end plate potential initiated? So here's our myelinated axon coming down. Say it's propagating an action potential, and that action potential arrives at the synaptic knob at the neuromuscular junction. That action potential is going to open voltage-gated calcium channels. Now, calcium, there's a higher concentration of calcium in the extracellular fluid, so calcium is going to influx into the synaptic knob or the axon terminal. That influx of calcium causes these synaptic vesicles, which are present and they lie in waiting. That influx of calcium causes the docking and exocytosis of these vesicles. So we get the dumping of acetylcholine into the synaptic cleft. That acetylcholine is going to very quickly diffuse across the synaptic cleft, bind to the acetylcholine receptors, and open them. And when they're open, then we have sodium, which will influx into the into the skeletal muscle fiber cell, and that's going to cause a local depolarization, that end plate potential. And if that's great enough, if there's a great enough depolarization, that's going to open up voltage-gated sodium channels, and then we can get an action potential propagated along the length of that sarcolemma, of the skeletal muscle cell. And so these steps, you should know these steps. Now, so we have skeletal muscle cells, which are pretty long and cylindrical, and they're just jam-packed with myofibrils, those arrangements of actin and myosin. And we need to, in order to get like maximum muscle contraction, we need all of those myofibrils to undergo contraction. So how do we get this action potential that starts in just Remember, there's only one axon terminal that synapses with one skeletal muscle fiber cell, but these muscle fiber cells can be quite long. So how do we get an action potential that can propagate along the entire length of the sarcolemma, but also penetrate into the muscle fiber to get all of those myofibrils to contract? And that's because skeletal muscle cells have a very extensive transverse tubule-sarcoplasmic reticulum system. And I'll go over these. I just have some bullet points as far as what they are. T-tubules are invaginations of the plasma membrane itself, and I'll show you an image of what this looks like. And then the sarcoplasmic reticulum, which skeletal muscle fiber cells have very extensive sarcoplasmic reticulum, is very similar to endoplasmic reticulum, and it's specialized in the fact that it can store large amounts of calcium ions. So keep that in mind. So here, this is just showing you lots of myofibrils, so these bundles of actin and myosin arrangements, which I think you got from a previous lecture. So we have all of these myofibrils, and this is our sarcolemma, or the plasma membrane. And that sarcolemma invaginates down into this tubular system, and these are T-tubules, which are literally just invaginations of the plasma, extensions of the plasma membrane. And in the center of these T-tubules, it's just going to be extracellular fluid, all right? So that's what a T-tubule is, invaginations of the plasma membrane. Now, right next to the plasma membrane are these T-tubules of the plasma membrane. Right next to them, you have these expansions of the sarcoplasmic reticulum, and these are called terminal cisternae of the sarcoplasmic reticulum. So they are right next to the T-tubule systems, okay? And you can see that this T-tubule sarcoplasmic reticulum system is quite extensive and goes around all of these myofibrils. And so together, the transverse tubule and the two abutting terminal cisternae are referred to as the triad. Okay. All right, so when I talked about how, acetylcholine is going to get released from the axon terminal, and it's going to diffuse across the synapse, it's going to bind to acetylcholine receptors, open them up, we get sodium influx into the skeletal muscle cell, we get an end plate potential, and if that depolarization is great enough, we get opening of voltage-gated sodium channels and an action potential on that skeletal muscle membrane, right? Well, the action potential is going to travel down all of the plasma membrane of that skeletal muscle cell, including those invaginations called T-tubules, because again, these are just invaginations of the plasma membrane. And so the action potential travels down these T-tubules, and it's going to affect receptors, or channels rather, that are connected to the sarcoplasmic reticulum. And so this is what I'm going to show you now. Here's our skeletal muscle membrane. That action potential is going to travel down the length of the plasma membrane of the skeletal muscle cell and also down the T-tubules. There are these special receptor channels called dihydropyridine receptors, DHP receptors, fine for you to know. But these guys are connected to calcium channels on the sarcoplasmic reticulum. And remember that the terminal cisternae of the sarcoplasmic reticulum they're right next to the T-tubules. So when that action potential comes down the T-tubules, it's going to affect these DHP receptors, causing these calcium channels to open, and there's way more calcium stored in the sarcoplasmic reticulum than what is present in the cytoplasm of the skeletal muscle cell. So when there's an action potential, these calcium channels get open, and calcium is just going to flood out into the cytoplasm of those skeletal muscle cell. And that calcium is then going to go and bind to troponin. And I believe you got this, like the actin, myosin, cross-bridge cycle from one of your previous lectures. But we get that calcium released from the sarcoplasmic reticulum. It can then go bind to troponin, which changes the conformation of tropomyosin, exposes the myosin binding sites on actin, and then we can get that cross-bridge cycling happening, okay, that cross-bridge cycle to happen. And that's how we get skeletal muscle contraction. I know this is somewhat small to see. I don't expect you to be able to read these words. I just wanted to show you another way to look at this. Here is our synaptic knob with our acetylcholine vesicles, our vesicles containing acetylcholine. We have an action potential arrive at the synaptic knob. We get voltage-gated calcium channels opening. Calcium is going to rush into the synaptic knob. That, in turn, that rise in calcium is going to cause exocytosis of these synaptic vesicles, the dumping of acetylcholine into the synapse. Acetylcholine is going to diffuse across, bind to acetylcholine receptors, change their conformation, cause them to open. Now that the acetylcholine receptors are open, sodium can influx into the skeletal muscle cell, causing an end plate potential, so a local depolarization. If that depolarization is great enough, we will get opening of voltage-gated sodium channels. And then just like you learned in your previous lecture, what happens along an axon, we get an action potential that propagates along the entire length of the sarcolemma, that plasma membrane of the skeletal muscle cell. And that action potential is also it's going to propagate again along the entire length of the sarcolemma, including down those T-tubules. Those special dihydropyridine or DHP receptors are affected by a voltage change, like an action potential. They're connected to calcium channels on the sarcoplasmic reticulum, which is right next to the T-tubules. So those dihydropyridine receptor channels are going to open up calcium channels. Calcium is going to flood out into the cytoplasm of the skeletal muscle cell and go bind troponin, and now we can get skeletal muscle contraction. Now we need skeletal muscle contraction to end at some point. We don't want our muscles to be contracted forever. And the neuromuscular junction with skeletal muscle cells is really efficient in removing acetylcholine. So there's lots of this enzyme present in the synapse called acetylcholine esterase. And acetylcholine esterase very, very quickly, acetylcholine only kind of hangs around if maybe a few milliseconds at most, because acetylcholine esterase, which is always present, will very, very quickly split acetylcholine into acetate and choline, all right? A lot of that choline will then end up being recycled back into the synaptic knob so that it can be reused to synthesize more acetylcholine that will then be packaged into synaptic vesicles. So a lot of it will be, we can have this choline being recycled back into the synaptic knob. We can also have acetylcholine or the breakdown products just diffusing away from the synapse. But we have to have that removed in order for that action potential to stop being propagated and for the skeletal muscle to be able to relax. Now here's this is a slide talking about, it just gives you some of the drugs that either enhance the action at the neuromuscular junction by drugs that kind of mimic acetylcholine and are able to bind to acetylcholine receptors and act similarly. Drugs that can enhance by inactivating acetylcholine esterase. So if we inactivate the enzyme that degrades acetylcholine, acetylcholine hangs around in the synapse more and we can get more muscle contraction. That's why it enhances the transmission at the neuromuscular junction. Or drugs that can block transmission at the neuromuscular junction. I'm not going to ask you to remember these drugs, but what I could do is I've talked about the mechanism that happens at the neuromuscular junction. I could give you a multiple choice question where I might give you a scenario, I'll mention a drug and what it enhances or what it inhibits, and then you would have to pick the, I'll give you choices A through D or whatever, and you might have to be able to tell me what would happen as a result of that drug being administered. So if you know the mechanism, if you know the steps that are happening at the neuromuscular junction, if I give you a drug that enhances or inhibits a certain part of that pathway, you should be able to tell me what will or will not happen next. I hope that makes sense. Let me know if that doesn't. This is just a clinical example of a chronic autoimmune neuromuscular disease. It's Myasthenia Gravis. And what happens here is that the immune system generates antibodies directed against the acetylcholine receptor. And so you have fewer acetylcholine receptors that are able to act appropriately. Fewer acetylcholine receptors means that you could have less muscle contraction in certain areas. So that's why you see these individuals will have drooping of eyelids, blurred or double vision, issues with facial expression, difficulty swallowing, anything that's going to really involve skeletal muscle contraction. But one of the ways that myasthenia gravis can be treated is by administration of an acetylcholine esterase inhibiting drug. And again, back to that mechanism or that pathway that we just talked about, is if you if you inhibit the enzyme that's going to break down acetylcholine, you'll have larger than normal amounts of acetylcholine present and accumulating in the synaptic space, leading to a greater likelihood of acetylcholine binding to the receptors that are functioning properly and getting appropriate muscle contraction. This is another slide. I'm just highlighting some different drugs that either inhibit certain, so for example, this conotoxin, it's got a negative sign next to it. So it's going to inhibit those voltage-gated calcium channels. So if I gave you a question involving, you might have to apply, I'll tell you that conotoxin inhibits voltage-gated calcium channels on the synaptic knob. And then you might have to apply that based on the fact that you know the pathway. What would not be able to happen next, right? What would that cause? And if you're inhibiting these voltage-gated calcium channels, we can't get the influx of calcium into the synaptic knob, and what would happen as a result would be we could not get exocytosis of these synaptic vesicles. So acetylcholine could not be released into the synaptic cleft. So I could give you a situation where you have to apply what you know and tell me what would happen as a result of inhibition or activation of the certain components of the pathway that you just learned, okay? So I'm not gonna ask you to remember all of these drugs. I'll tell you the drug, I'll tell you what they activate or inhibit, and then you apply that, you use the knowledge that you know about this pathway to apply that and answer my question.